This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So now we are learning about the second chapter, biological classification, and we have done a quick review about all the kingdoms, kingdom Protista, kingdom Monera, fungi, Animalia, Plantae, about all these kingdoms we have learned. And now we are solving the previous year question papers in this part. So already we have done around 42 questions. So here uh, we have to learn about all the main five groups or kingdoms, and we have to learn the main features of that. And in this chapter we are learning about kingdom Monera, Protista, and fungi mainly, and along with that about lichens, viroids, and viruses also we are learning, right? So let's start with the question number 43. Up to that we have already done. So in this question, we said what is true for cyanobacteria? Whether they are, first option is oxygenic with nitrogenase, oxygenic without nitrogenase, non-oxygenic with nitrogenase, and non-oxygenic without nitrogenase. So here we should know what is cyanobacteria, right? Question number 43. You're solving question number 43. So we should know what is cyanobacteria. So cyanobacteria, we have already learned, that is also called as blue-green algae, and it's a very important group of photosynthetic bacteria, right? It is having photosynthetic ability. So it is a photosynthetic bacteria, and the cyanobacteria, we know that photosynthetic bacteria, and that can fix atmospheric nitrogen with the help of one enzyme. So that can fix atmospheric nitrogen, and that fixation is done with the help of enzyme nitrogenase. With the help of which enzyme is involved in that? Nitrogenase, right? And this is showing which type of photosynthesis? Oxygenic photosynthesis. Which one? This cyanobacteria. So here among the four options, which option is correct? Option A, oxygenic with nitrogenase. So here you can see the presence of nitrogenase enzyme, and this is an oxygenic bacteria, or it is doing the oxygenic photosynthesis. Clear? So that's how you need to choose the option. Then go to question number 44. Here in question number 44, you can see what is true for photolithotrophs. Okay, about photolithotrophs. So photolithotrophs, they're using light as energy, right? So when we are saying about photolithotrophs, so photolithotrophs used light as energy. Here light is energy and inorganic electron donor. From where it is getting hydrogen source? Hydrogen source, inorganic electron donor as hydrogen source. Right. So inorganic electron donor means that may be hydrogen sulfide or something like that. That will be the source of hydrogen. Okay. So hydrogen ions that will come from this inorganic electron donor. Right. And light is the source of energy. Then photolithotrophs examples. example, we here while learning about this question, while discussing about this question, I'll say the examples also. Like they are purple and green sulfur bacteria. Purple and green sulfur bacteria. They are the examples for photolithotropes. So photolithotropes, you can say here the right answer, here options. They are obtaining energy from radiations and hydrogen from organic compounds. Second option is obtaining energy from radiations and hydrogen from inorganic compounds. And third option, they are obtaining energy from organic compounds. Fourth option, they are obtaining energy from inorganic compounds. Here, actually, here we said from inorganic compounds, they are getting the which one? Electron donor. So this option, these both options, obtaining energy from organic compounds, obtaining energy from inorganic compounds, no. They're the organic or inorganic compound that is working as the provider of electron, right? So that two options we can cancel. Then we can go for option A and B. And here option A says that energy from radiations. That part is correct, no doubt in that. They are getting energy from radiations, light. That is energy source, right? And hydrogen from organic compounds. No, they are getting hydrogen from, here we said they are getting hydrogen from inorganic or the electron donor, that is inorganic compound. So option B is the right answer here. Obtain energy from radiations and hydrogen from inorganic compounds. That's the right answer. Option B is the right answer in the case of photolithotropes. Right. So next we are moving to question number 45. In question number 45, you can see photosynthetic bacteria have pigments in. Option A is leucoplast, option B is chloroplast, option C is chromoplast, option D is chromatophores. Right? These are the options. So here we know that in photosynthetic bacteria, in photosynthetic bacteria, we know photosynthetic bacteria, they have small particles, right? Small particles which are around 60 micrometer in diameter, which are called as chromatophore. These small particles are called as chromatophore. Right, these are the small particle which is around 60 micrometer in diameter. They are called as chromatophores and they are present. So these chromatophores are attached to the inner surface of the cell membrane. Right, where these chromatophores are present? Inner surface of cell membrane. Right, in plants we can say that in photosynthetic plants they have pigments or chlorophyll. In chlorophyll they can get all these things, right? So chlorophyll is helping for photosynthesis. What about in the case of bacteria? They have very small particles which are around 60 micrometer in diameter, which are called as chromatophore, and these chromatophores are present in the inner surface of cell membrane. Okay, in the inner surface of cell membrane these chromatophores are present, and that works as bacteriochlorophyll. Okay, this serves as the bacteriochlorophyll. Which one? Chromatophore. Remember that name, chromatophore, which is the chromatophore, which is a very small particle around 60 micrometer in diameter, and that is present in the inner surface of cell membrane. These are attached to the inner surface of cell membrane. Clear? And this is working as a bacteriochlorophyll. right? Next question is question number 46. 46th question. A few organisms are known to grow and multiply at temperature of 100 to 105 degree centigrade. They belong to, first option is marine archaebacteria, option B is thermophilic sulfur bacteria, option C hot spring blue-green algae, and option D thermophilic subaerial fungi. These are the four options. So here it's a very direct question, like the organism which are known to grow and multiply at temperature of 100 to 105. They are thermophilic because they have a positive tendencies towards the higher temperature, right? So they are thermophilic. So here, which is the right answer? Option B, thermophilic sulfur bacteria. Okay, thermophilic sulfur bacteria. That is the right answer. And from here, the question itself, we are getting some organisms like thermophilic sulfur bacteria. They are the organism which can grow and multiply at temperature of, what is their temperature range? 100 to 105. Right? They are belonging to thermophilic sulfur bacteria. Right? So such kind of direct questions are coming means there no need to take even 1 minute. You can save the time in this kind of questions. 47. Question number 47. Here, the site of respiration in bacteria. See, the site of options they have given us episomes, mesosomes, ribosomes, microsomes. Okay, so among these you need to select one. So we know that the cytoplasmic membrane of bacteria, in the case of bacteria, cytoplasmic membrane, that is having invaginations or inside foldings at some places which is into the cytoplasm, right? So it is having inside foldings into the cytoplasm, and that is in the form of tubules. Invaginations like tubules inside, towards inside the cytoplasm, right? These are called as, what is the name of this? Mesosomes. Mesosomes. And on their surface, the enzymes which are related to respiration that are associated. So mesosomes work as mitochondria in the case of bacterial cell, right? Cell respiration, which is the site of cell respiration, mitochondria. Here the mesosome is working as the mitochondria in the case of a bacterial cell. So in the bacterial cell, which is playing the role of mitochondria here, mesosomes. Okay. Next question number 48. Question number 48. In bacterial chromosome, the nucleic acid polymers are option A, linear DNA molecule, option B, circular DNA molecule, option C, of two types DNA and RNA, option D, linear RNA molecule. So in bacterial chromosome, nucleic acid polymers are, so we know that in, hope you have heard about nucleoid, we have learned. This nucleoid or the chromatin body, it is otherwise called as chromatin body, or we can say that genophore. Genophore. So this, where it is present in bacteria, right? So this genophore that occupies around 10 to 20% of cell, right? This is around 10 to 20% of cell, and which is present near the center of the cell, right? This is present in the center of the cell. And here in the genophore or nucleoid or chromatin body, you can see that consists of a single circular DNA molecule. That is a single circular DNA molecule, and all genes are linked in this, right? So in which all the genes are linked, and this is around 1,000 times longer than the cell itself, but it is highly folded inside, and it is therefore folded in a very higher way, right? Because it is many times longer than a cell, right? And that doesn't have histone proteins. Histone proteins are absent. About which one we are saying? We are saying about the bacterial chromosome. So here the option B says it's a circular DNA molecule. Right, we said some more points. The nucleoid or chromatin body or genophore in bacteria, that is the bacterial chromosome part. It is 10 to 20 percentage portion it is covering in the cell. So in the bacteria cell, it covers around 10 to 20 percentage of the cell, and it is present in the center. And this is a single circular DNA molecule. Okay, so here we have got an option, circular DNA molecule. This is a single circular DNA molecule, and here histone proteins are absent. And this is highly folded, and that occupies 10 to 20 percentage of that bacterial cell, and all the genes are present on that, and that is many times longer than the cell itself. That's why it is highly folded. Clear? So option B is the correct answer. Next, question number 49 here. Sex factor in bacteria is option A, chromosomal replicon, option B, F replicon, option C, RNA, and D, sex phylus. 49, question number 49. So here bacterial strains, we know that bacterial strains like E. coli, that and all show sexual differences. You can see such type of bacteria will show sexual differences. So each male cell that is having a sex factor or the fertility factor, which is called as F factor. Okay, so in that male cell, male bacterial cell or bacterial strain, which is in some kind of bacterial strain, they are showing sexual differences, and in male cell they will be having a sex factor, or they'll be having a sex factor, or we can say that as a fertility factor. So this fertility factor, in short, we can call as F factor. F factor or fertility factor, right? And this F factor or fertility factor, this is a small piece of DNA which is in circular form. F factor, that is a small circular DNA. Okay, so it's a small circular piece of DNA, and this is having the capacity of self-replication. This is self-replicating small circular DNA, but that is having 1 out of 100, that much only it is having size. Okay, so here what answer we'll get finally? Which one we can select from these options? 49, sex factor in bacteria is option B, F replicon. Right. So which is that F factor or F replicon that is called as a sex factor in bacteria. Right. Next, the question number 50. Question number 50, you can say the plasmid. About plasmid. This question is about plasmid, and here you are getting 4 options. Plasmid helps in respiration, genes found inside nucleus, and plasmid is a component of cell wall of bacteria, is the genetic part in addition to DNA in microorganism. Right? So plasmid, we know what is plasmid. Plasmid is a very small self-replicating extra chromosomal material in bacteria. This is small. Plasmid is small. Then it is having the capacity of self-replication. Then it is chromosomal, extra chromosomal. It's an extra chromosomal material and it's a non-essential genetic element. Non-essential genetic element, right? Already genetic element will be there, and that chromosome, apart from that chromosome, this is an extra chromosomal material, and this is very small and it is having self-replicating ability. Okay, so plasmids are small and self-replicating extra chromosomal non-essential genetic element in bacteria, right? And each plasmid, we can say, that has a ring. Each plasmid that has a ring of circular, supercoiled, then double-stranded DNA, right? And they are carrying the genes for replication for one or more cellular non-essential functions. Okay, so these cellular non-essential functions, for that it is coding for, and these are also called as mini chromosomes, and it is otherwise called as dispensable autonomous elements. Okay, so these are the extra points here. Which option you need to select? 50, question number 50, which option you need to select? Option D. It is a genetic part in addition to DNA in microorganism. We said that is in bacteria. This plasmid we can say in microorganism, and that is a genetic part which is additional to the genetic material of that particular microorganism. So it is addition to DNA in microorganism. Already DNA will be there. Additionally, this plasmid also will be there. It is very small and it is having self-replicating capacity. It is extra chromosomal. Apart from the chromosomal material of that organism, this is an extra chromosomal one and non-essential genetic element. This is not an essential genetic part. It is non-essential genetic element which will be doing some extra functions, like it is coding for non-essential functions, right? It's not the must one, but it may code for non-essential functions, right? So here option D is the right answer. Next, question number 51. Question number 51. Temperature tolerance of thermal blue-green algae is due to, here you are getting 4 options: cell wall structure, then cell organization, mitochondrial structure, homopolar bonds in their protein. So here you know that cyanobacteria, that is providing a very best or very good example of the adaptability of life to the extreme conditions, right? Cyanobacteria. Cyanobacteria is otherwise called as blue-green algae. So they can live in the extreme conditions, and that shows their adaptability of life to the extreme environmental conditions. that means high temperature of hot springs and low temperature of polar region, everywhere they can exist, right? So this is due to their gelatinous sheath, which can withstand long period of desiccation, right? So how is it possible? How these extreme living conditions they can withstand? Because they have a gelatinous sheath. They have a gelatinous sheet, and that can withstand all these extreme conditions, right? So the compactness of protein molecules and their bonds in the protoplasm, compactness of protein molecules and their bonds which is present in the protoplasm, that is helping them to face all these extreme conditions, right? So here option A, cell wall structure, that is the right answer. So cyanobacteria, they are having good adaptability to live in the extreme conditions, and that is due to the molecules, protein molecules which are present in their protoplasm, and their compactness, the compactness of this protein which can survive in the extreme conditions. So here the right answer is answer A, cell wall structure. Structure of cell wall is helping them to sustain or to help to withstand these extreme conditions. Okay. Then question number 52. Which of the following cannot fix nitrogen? Question number 52. Here, nostoc, anabina, azotobacter, and spirogyra. These are the options, right? Option A is nostoc, option B azotobacter, option C spirogyra, option D anabina. Here you know that spirogyra cannot fix atmospheric nitrogen because it is a member of green algae, isn't it? Spirogyra. Here the answer C, spirogyra, that is the one which cannot fix atmospheric nitrogen, right? That cannot fix atmospheric nitrogen because that is belonging to green algae, green algae, right? So here only nostoc and anabina and azotobacter. Nostoc and anabina both are, which one? Nostoc and anabina both are blue-green algae. And azotobacter, that is a bacteria which that also can fix atmospheric nitrogen. So that can fix the nitrogen. And nostoc and anabina already they are cyanobacteria or blue-green algae. They can fix atmospheric nitrogen. Here the one and only option which cannot fix atmospheric nitrogen is spirogyra. Clear? Next question, question number 53. Nitrogen fixer soil organism belong to? See, just now we said nitrogen fixing organisms, which are they? Nostoc, anabina, such organisms are there. So nitrogen fixer soil organism like Acidobacter, Rhizobium frankia, all these are belonging to bacteria, soil organisms, especially about soil organisms they are asking. So the nitrogen fixing soil organisms, here you are getting options mosses, bacteria, green algae, soil fungi. So which is that? Soil organism that is belonging to group bacteria. Okay, so which are the examples? Acidobacter, Rhizobium frankia, all these are belonging to bacteria. Then question number 54. Question number 54. E. coli is used extensively in biological research as it is option A, easily cultured, and option B, easily available, option C, easy to handle, option D, easily multiplied in host. So here, Escherichia coli or E. coli, that is a commensal bacterium, and it is most studied bacterium, and that is widely used in researches because it is easy to culture on a minimal medium, right? And the fast rate of multiplication is there, and the generation time is also very short. That's why researchers are using that. Researchers are using because it can be easily cultured, right? So how, if we can culture that easily, means they are widely accepted for all the studies, and their multiplication time will be less, and within short time it can be cultured, and can be cultured in minimal medium requirement and fast rate of multiplication. Everything is a reason. So that's why it's easily cultured. That's the best option. It is easy to handle, easily available, easily multiplied in host. Apart from all these things, the best option is easily cultured, right? For culturing equally, it's very easy. Next question number 55. Gonophore bacterial genome or nucleoid that is made of, first option, histone and non-histone, option B is RNA and histone, option C is single double stranded DNA, and option D is single stranded DNA. So here, genophore bacterial genome or nucleoid. So in bacteria, nucleoid or genophore is haploid, and that consists of a supercoiled DNA, which is highly folded, and that is a circular ring-like DNA, and that is double-stranded, and it is a single and naked DNA. So all these characters you can see, and there is no free end and no histone protein also, right? So here question number 55, that is a single double-stranded DNA. Option C is the right answer here, because histones and non-histones, RNA and histones, single-stranded DNA. No, it's not a single-stranded DNA. So here it is a single double-stranded DNA. Clear? So option C is the right answer. Next question number 56. Which one of the following organisms are known as chief producers in the ocean? So in the ocean, you're getting which are the chief producers in ocean. First option is dinoflagellate, option B diatoms, option C euglenoids, and option D cyanobacteria. So diatoms are the chief producers in the ocean, and they contribute. Question number 56. Chief producers are diatoms in the ocean, and they are contributing around 40% of marine primary productivity, and they constitute, or they are forming, a major group of unicellular eukaryotic microalgae, right? Unicellular eukaryotic microalgae, and among that the most common type is phytoplanktons, right? And the here you can say the diatoms, they are the chief producers of the ocean, right? and they are contributing around 40% of marine primary productivity. So in the 56th question, here you are getting option B. Chief producers are diatoms. So these are the direct kind of questions. So here it won't take you more than 10 to 20 seconds to select the answer, right? Next question, number 57. Ciliates differ from all other protozoans in options are using pseudopodia for capturing prey, option B having a contractile vacuole for removing excess water, option C using flagella for locomotion, option D having two types of nuclei. Right. So here ciliates, here we know that ciliates, they are different from all other protozoans and they having two types of nuclei, right? They differ from other protozoans because they have two types of nuclei, right? Two types of nuclei. Right? And these two nuclei, they are of different size. That is, one is meganucleus and the other one is micronucleus. One will be meganucleus and other one will be micronucleus. And the meganucleus, the first one, the meganucleus that controls the metabolism, and the micronucleus that is connected with the reproductions, right? So for example, you can say um, ciliates. For that example, paramecium. Paramecium, you can see several cilia around that, right? So here, two types of nuclei you can see. That's why the ciliates are different from other organisms because they have two types of nuclei. One is, both are having different sizes. One is meganucleus and the other one is micronucleus, and the meganucleus that is controlling the metabolism and the micronucleus which is related with the reproduction. So example is paramecium. Okay, so here which answer we can select? Ciliates differ from all other protozoans. The option is they have two types of nuclei. We said they have two types of nuclei, so they are different from the normal organism, right? And question number 58. Chrysophyte, euglenoids, dinoflagellates, and slime molds, they are included in the kingdom. Which kingdom? So all these single-celled eukaryotic organisms like chrysophytes, euglenoids, dinoflagellates, and slime molds, all these are included in kingdom Protista, right? Kingdom Protista, kingdom fungi, kingdom Animalia, and Monera. So among that, the ciliated organism, they are working or they are coming under kingdom Protista. Option A is the right answer. Then question number 59, select the wrong statement. First option is the walls of diatoms are easily destructible. Option D, diatomaceous earth is formed by the cell wall of diatoms. Option C, diatoms are chief producers in ocean. Option D, diatoms are microscopic and float passively in water. Right. So here you can see question number 59. So in this 59th question, which is a wrong statement? The wall of the diatoms are easily destructible. That is a wrong statement. Right? It's not so easily destructible. See, diatoms, they are single-celled and plant-like protists, and that produce intricately structured cell walls, and that is made of nanoparticles like silica and all. So the walls are indestructible. The first option itself is wrong. Walls of diatoms, they are indestructible. So the right answer is the walls of diatoms are easily, the walls of the diatoms are easily destructible. So the statement option A is the incorrect one. It's not easily destructible, it is indestructible. So first option itself they have given the walls of the diatoms are easily destructible. So that is a wrong answer. Their walls of diatoms, they are easily indestructible. Okay, we cannot destruct that easily. So that is the wrong statement among the four options, right? And question number 60, if you are moving to question number 60, pick up the wrong statement. Again, you have to pick up the wrong statement. Okay, so in this you can get the cell wall is absent in animalia. Second option, protists have photosynthetic and heterotrophic mode of nutrition. Option C, some fungi are edible. Option D, nuclear membrane is present in Monera, right? See here, in animalia cell wall is absent, there is no cell wall, right, in animal kingdom. That is correct statement. And protista have photosynthetic and heterotrophic mode of nutrition, that is also right. And some fungi are edible because mushrooms are coming under fungi kingdom, fungi, so they are edible, that is also correct answer. Next, option D, nuclear membrane is present in Monera. See here, option D is incorrect, right? Nuclear membrane is present in Monera. And how we can correct that statement? See, Monera, that is a kingdom which is having unicellular organism, and they have the prokaryotic cell organization. That means they lack nuclear membrane and their membrane bound organelles. They don't have nuclear membrane and membrane bound organelles. So there we are staying. See, we here in the option D, nuclear membrane is present in Monera, that statement is incorrect, right? So correct option is option D. And next question, in which group of organisms the cell wall form two thin overlapping shells which are fit together? See question number 61. See here, options chrysophytes, the nucleinoids, dinoflagellates, and slim modes. Here the chrysophytes, question number 61, we need to say that here chrysophytes, they are placed under which kingdom? Kingdom Protista. Chrysophytes are coming under kingdom Protista. They are coming under kingdom Protista, right? And this group, they are including diatoms and golden green, or we can say desmids. So most of them are photosynthetic. And in diatoms, the cell walls that form two thin overlapping cells, and they are fit together like a soap box. In a soap box, how the two overlapping cells are fit together, like that it will be there. In which one? In diatoms. In diatoms, the cell wall form two thin overlapping cells, and that can be fit together as it is in the, as we can see in a soap box, right? So here the right answer of 61 or question number 61, that is chrysophytes. Okay, these there is no overlapping shells which are fit together in the slimmodes, euglenoids, and dinoflagellates and all. The right answer is chrysophytes. Clear? So one more class we need to discuss about these questions to complete the questions from this chapter. So in the next class, we'll be learning about few more questions about kingdom protista we'll solve, then kingdom fungi, then about virus and viroids we'll discuss. So all those questions in virus and viroids, mostly you will get direct questions. And here again confusion, little bit confusion will come in kingdom fungi. Okay, so if you are answering these many questions, there is nothing to learn extra from this chapter. So like this, you will get first and second chapter, which you can cover very easily, because there is no so many application type of questions. Almost you will get direct questions only. Okay, thank you.